We have been probing around up here by the cross hole, and just immediately to the left of the film container you see some dark pigmented material. Now this appears to be dried blood, but we do plan to put some of this in the film case and take it back and have it analyzed to see if it is blood. There, right at the very tip of the tab, there's some more of this real dark material. As soon as it's exposed to the light, it begins to turn a lighter brown, and this is typical of very old blood. So we'll take some more of this sample here, and it seems to have come down through this area here, as we've collected it all the way along and down into this crevice that communicates with the chamber that the Ark of the Covenant's in. Anyway, I have several samples of it that I have taken.
For many years, the specimen sat untouched. Then, in 1996, when Ron was in Great Britain, Richard and Elizabeth Reeves and their sons came to Nashville to our home. Richard had a new, unique, and very powerful microscope he wanted to show us.
Leviticus 17:14 informs us that the life of all flesh is the blood thereof. Once again, scientists are finding out that the truths of Scripture are far more literal than they ever imagined. Tiny particles have been discovered in blood that are seemingly indestructible. These tiny particles are less than 1/10 of a micron in size, and while they cannot be seen using standard medical microscopes, new methods of microscopy have been developed which makes it possible to see them in their living state. The French scientist Gaston Naessens designed special microscope components that would allow him to see these particles, which he entitled somatids, meaning the body that creates. Through experimentation, Professor Naessens found that these somatids contain genetic material yet to be fully understood by the scientific community. They are polymorphic in nature, growing from stage to stage, and are unaffected by high temperatures, extremely toxic chemicals, or even nuclear radiation. Professor Naessens believes these somatids to be subcellular living and reproducing entities, the precursor of DNA, and that they may be the building blocks of life. About 6 years ago, I learned about these particles as a result of some research that I was conducting, which was totally non-related to archaeology. At that time, I was using a standard medical microscope, and while I could not see these particles, I knew they had to be there. I began to develop my own microscope patterned after the design of Professor Naessens. What you see behind me is that microscope in its latest stage of development. The microscope employs an extremely bright xenon or mercury light source, which can be combined with tungsten light. The light is directed across the microscopic specimen rather than on or through it in what is known as a dark field configuration. The light reflecting off the object is transmitted through the microscope and is magnified over 1,000 times. The image is then captured by a sensitive CCD camera and transmitted to a monitor for viewing at screen magnifications of up to 30,000x. Under these conditions, the tiny living particles can plainly be seen against a dark background. In 1996, my family and I made a trip to Nashville to visit the Wyatts. I brought along the microscope, which at that time was somewhat portable in its early stage of development, and set it up in the basement of the Wyatt's house. One evening, Mary Nell Wyatt asked me to take a look at some material from a burial cave to see if these tiny particles were present. Without my knowledge, one of the samples was actually the blood sample that Ron had taken from the Ark of the Covenant dig. The sample was placed under the microscope, and as the specimen began to come into focus, thousands of tiny particles, somatids if you will, became plainly visible. At that time, Mary Nell, who was standing behind me, began to weep. As I turned around and saw the expression on her face, I realized immediately that the sample we were looking at was actually the sample that Ron had found to be the blood of Christ. Okay, Mary Nell's going to take the sample and mix it with sterile water. That'll be enough. Okay. And she's going to take some sterile water. And just some drops, all we need, Mary Nell. Okay, so now that we've got the sample on the slide, we'll go ahead and focus into the sample and see what comes up on the screen here. And there they are, thousands of living particles, less than a quarter of a micron in size. The spores are maybe a half micron, and but most of those little particles that you're looking at is 17,000 X, magnified on the screen 17,000 times. You know, in the Bible it tells us that the life is in the blood, and there's little living particles that most people are unaware that are there.
The results we obtained in no way can be said to be proof that the specimen was the blood of Christ, but based on the location from which the specimen came, we believe it was what Ron believed it was. Years earlier, Ron had taken a blood specimen from the lid of the case which contained the Ark of the Covenant.
The blood of Christ is only dried out, folks. It's not dead. When we rehydrated it with normal saline, 72 hours at body temperature with slight, very gentle swirling, and put the white blood cells in a growth medium, 48 hours later, we did a chromosome count. I didn't. This was in Israel. It has 24 chromosomes only. All of us here have 46. Unless, you know, there's a couple of genetic anomalies that make that different. But Christ received 23 from his mother and one Y, sex determining factor, from his father, who was not a human father. Because had he received that from a human father, it would have been accompanied by 22 autosomes. Now what this basically means is that his height, his eye color, his hair color, and all of this was supplied from the genes of his mother's gene pool. However, Mary and Joseph both descended from David, but none of us have 24 chromosomes. Well, they knew. They told me before I asked them when I asked them to perform this investigation, that dried blood, you can't get a chromosome count on it because the white blood cells have to be alive and well in order to do that. You can get DNA, you can get some other things, but you can't have a chromosome count. So this blood is unique, and it is Christ's blood.
Science has finally unlocked a secret hidden for 2 millennia. By sequencing the DNA from relics tied to Jesus, researchers thought they would close a chapter of history. Instead, they found a genetic code that's not in any textbook. Buried in the data is a sequence so bizarre, so alien, it has sent shockwaves through the scientific community. You see, the DNA is human, but only partly. The rest is something else entirely, a biological signature that's frankly inhuman. Forget everything you thought you knew. This discovery redefines the boundaries of life itself. A 2,000-year-old mystery. Recently, a quiet, secretive project gave a handful of scientists permission to do what was once unthinkable, to take samples from these priceless relics and sequence the DNA within. This wasn't just a simple history project, it was an attempt to decode the genetic identity of the most famous person who ever lived. The challenges, to put it mildly, were monumental. You see, DNA is a fragile molecule. After just 500 years, about half of it is gone. After a couple of thousand years, what's left is shattered into tiny, almost unreadable fragments. And that's before you even consider contamination. For 2,000 years, these cloths have been touched, kissed, and wept on by countless pilgrims, popes, and kings. Every touch left behind a genetic fingerprint, creating a storm of DNA from thousands of different people across Europe, the Middle East, and beyond. It's like trying to find one specific grain of sand in the middle of a hurricane. The team used cutting-edge techniques, the kind of technology usually reserved for solving modern crimes or studying the bones of Neanderthals. They isolated the tiniest threads, soaked in what appeared to be real blood. They targeted specific areas where the original material was most likely to be preserved, away from the edges that had been handled the most. Their goal was to find a consistent genetic profile deep within the fibers, a single voice that could rise above the noise of centuries. They were looking for a male of Middle Eastern descent with genetic markers that would place him in the Levant 2,000 years ago. What they found started out exactly as they predicted, but then as the data kept pouring in, a pattern emerged that made them question everything they thought they knew about human genetics. The most shocking fact is that this wasn't just about identifying a man. It was about discovering a biological signature unlike anything ever recorded. The first results confirmed the blood type was a B, rare worldwide but more common in the Middle East. They found Y chromosome markers confirming the subject was male and mitochondrial DNA pointing to a maternal line from the region. It all fit, but then they saw it, a series of genetic markers that simply didn't belong, sequences that had no match in any global DNA database. It was human, yet at the same time, it was something more. Something else. The lab was on the verge of a discovery that could shake the foundations of both science and religion. Blueprint never seen before. Inside the clean rooms of the sequencing lab, supercomputers worked for days, piecing together the shattered puzzle of ancient DNA. The first layers of the genetic code were straightforward. They confirmed what many had long suspected: the man whose blood stained these cloths had roots in the Middle East. The specific haplogroups, the ancestral clans of our DNA, pointed directly to the area populated by Jewish communities 2 millennia ago. This was a huge win. It pushed back against the famous carbon dating test from 1988, which had controversially labeled the Shroud a medieval fake. But what many overlooked in that initial excitement was the strange data coming from the Y chromosome, the part of our DNA passed down from father to son. You see, the Y chromosome is like a family tree for the male line. It carries a history of a man's ancestors stretching back thousands of generations. The scientists expected to find a common Middle Eastern lineage, one that would connect to known populations. Instead, they found a genetic island. The core of the Y chromosome was there, but it contained what are known as tandem repeats, long, stuttering sections of genetic code that were arranged in a pattern never before seen by science. It wasn't just a new branch on the human family tree. It was as if a whole new tree was growing right next to ours. It was recognizably human in its structure, yet completely alien in its details. One scientist described it as finding a book written in English, but with a dozen new letters in the alphabet. This was the first inhuman result, but it wasn't the last. The team then turned to the mitochondrial DNA, or mtDNA, which tells the story of the maternal line. Here too they found something that defied explanation. The primary haplogroup was H, common in Europe, but with a subclade H33 found among the Druze people of the Levant, a perfect fit for the region. But mixed in with it were fragments of another maternal line, one so ancient and distinct it looked like it had branched off from the rest of humanity not thousands but hundreds of thousands of years ago. It was closer to the genetic offshoots of early hominids like the Denisovans than to modern humans. And you can see this pattern repeated across the genome. One part familiar, one part completely unknown.